Hey everybody, welcome yet again another journal club brought to you by Life Extension Advocacy Foundation, LEAF. This is Dr. Oliver Medvedik here, and we have a special guest with us, Matthew Oki O'Connor from Sense, who is going to be doing an introduction and going over some research that's going to be coming up, but also going over an interesting review article that he's been putting together looking at a specific specific causative agent for aging. There's a lot of things that go wrong with aging, but in this review article, the focus is on one particular nasty oxidized chemical called 7-keto-cholesterol. And if you have not heard of 7-keto-cholesterol, you're certainly going to hear all about it on this session here. Cholesterol, as we all know, is a sterol. It's a lipid. It's an important molecule for pretty much many different functions in our body, not just to manufacture different hormones, but also it's present in all of our membranes, our lipid bilayers. It's there for lipid fluidity. So it is a necessary component of life. But cholesterol, things can go wrong with it. It can get oxidized, and once it gets oxidized, it could lead to a lot of conditions such as atherosclerosis and a lot of other things that could disrupt the cell membrane. So this is going to be the topic of our discussion, at least one of the topics, basically how this one particular bad oxidized variant of cholesterol, 7-keto-cholesterol, is responsible for a whole host of aging-related disorders and what we can potentially do about it. So I'm going to turn the floor over to Dr. Matthew Oki O'Connor. Welcome.
Hi, thank you, and thanks everyone, and sorry to be getting started late. So I wrote this article, and I wanted to write this article to accomplish several things. One was to educate myself and ourselves, my team, on the ins and outs of this molecule, its chemistry and its biology. Two, to draw attention in the field, in the biomedical field and in the aging field, to this molecule and help educate people on its importance and why it is both unique in terms of its properties and its importance, ways in which it is unique, and also to draw attention to this concept of damaged molecules that can accumulate in aging. And so the point of the article wasn't to try to claim that this is some magically horrible thing, and if we could just get rid of this one type of oxidized cholesterol that we would somehow cure aging, that this is one kind of molecular damage that accumulates, and that what we need to do is tackle these things often one at a time and hone in on one, hyper-focus on it, and find ways to remediate it. The review article is mostly just about the molecule rather than methods to remediate it. But of course, we at Sense Foundation and in the company that I'm spinning out of Sense Foundation, Underdog Pharmaceuticals, are actually trying to do something about it. But before we can do something about it, we need to understand what it is and how it works. So I guess normally for a journal club, I would go through, I would sort of base the discussion around the figures, which I think is appropriate here, although possibly less comprehensive than you would in a normal data paper. But I thought maybe we could walk through the figures and then field questions from people about the different topics that are covered in the article. So I don't know what people are seeing, if they can be looking at the article, or if I should start sharing that now.
Yeah, you should go ahead and
Mhm.
share the paper on the screen, Oki, and everybody viewing at home will be able to see it.
Okay.
There we go. That's it.
So this, as you can see, the paper is in the late stages of being published at Redox Biology. Just about every other day, I get an email from the copy editor saying that it's going to be published tomorrow, but we'll see. It's the pre-publication is already available on their website, as many of you may have seen. Unfortunately, it's not the final proofed version, which has been submitted and completed. It just isn't worked its way through the system. So let me go through these highlights. And the only thing that's missing from this draft, or the drafts that some of you have seen, or you may see some typos or some grammatical errors that were corrected later, but the figures are all accurate, although in some versions of the downloadable material, the figures are low resolution, and some of them it's high resolution. It's a little bit of a mess, but I think the versions that I'll be able to show you today are pretty good. So the main points from this paper is that 7-KC is unlike many forms of cholesterol, it is non-enzymatically produced. So if you start with cholesterol, and I'll show a figure on this in a couple of minutes, and then you react it with a free radical, an oxygen-free radical, the most common product is going to be 7-ketocholesterol. Many other oxysterols are not bad, they're not toxic, they're actually signaling molecules, and they're produced in very specific enzymatic pathways. And I just want to be careful to distinguish between the non-enzymatic damage molecule that is 7-ketocholesterol and some other oxysterols, and the other body of oxysterols that are produced on purpose, if you will, in biology. And those are not bad things. 7-KC is implicated in all kinds of diseases, aspects of aging that we'll get into. I believe that makes 7-KC a great target for medicine, and I want to make the case that it should be a target for medicine. So moving on from there, let me take kind of a big picture of where 7-KC comes from and what it leads to. Like I was just saying, everybody knows what free radicals are, I hope, in this group. Free radicals, of course, are implicated in many aspects of aging and disease. The way that interacts with 7-KC is that you have regular cholesterol, you react with an oxygen molecule, and you end up with 7-ketocholesterol. And the only difference between cholesterol and 7-ketocholesterol is this one extra oxygen molecule at the 7 position that is double bonded to cholesterol. 7-ketocholesterol is bad because it looks and acts enough like cholesterol that it sort of impersonates cholesterol, but it cannot be used and metabolized in the productive ways that cholesterol can be used. So as Oliver was introducing, cholesterol is important for the fluidity of your cell membranes. But 7-ketocholesterol, when it replaces cholesterol in a membrane, changes that fluidity and makes it less fluid and more rigid. This can permeabilize membranes and damage both plasma membranes and internal membranes and internal organelles, such as lysosomes. So that means that if 7-KC gets taken up, it can be recognized as a damaged molecule and get shunted to the lysosome, but the lysosome is not well equipped to metabolize 7-keto cholesterol. So it tends to accumulate there and disrupt lysosomal function. There may be an interaction, and I talk about this in some depth in the article, between the mitochondria, which is a source of many of the free radicals, and the lysosome. But to what extent the oxygen radicals being produced by the mitochondria are creating 7-keto cholesterol in the mitochondria and then shunting them to the lysosome from there, or whether it's the free radicals leaking out of the mitochondria and reacting with cholesterol elsewhere, is still an open question. So I don't know whether these lines between the mitochondria and the lysosome are direct or indirect with regards to 7-KC, but of course there's well-known interaction between mitochondria and lysosomes, that being that when mitochondria become damaged and dysfunctional, they get eaten by lysosomes. So if 7-KC is being produced and accumulating in mitochondria, then that's a likely source of it. In terms of cells that are thought to accumulate 7-keto cholesterol, I give several examples, and this leads directly into several diseases. So the main one that I am dealing with in my lab is macrophages and their interaction with atherosclerosis. So what happens there is, just as we were just talking about, that macrophages, when they are going along and eating up junk, they can start eating 7-keto cholesterol and producing their own 7-keto cholesterol. When they accumulate too much of it in their lysosomes shut down and they will turn into foam cells. And I'll get into that more later. Microglia are kind of like the macrophages of the brain, and they are important for protecting you against Alzheimer's disease. And so they have the same problem. And so it is theorized, and not as well established as in the macrophages and atherosclerosis, but it's theorized that when the microglia get too much 7-keto cholesterol, that they have a hard time turning over and consuming the A-beta that can lead to the A-beta plaques, which would allow you to develop that canonical aspect of Alzheimer's disease. And the final example that I have here is in the eye, in the retinal pigment epithelial cells, which are supposed to eat up the shed rod outer segments of the rods in the eye, that they also can accumulate 7-keto cholesterol in their lysosomes, shutting those down and losing their ability to properly process those shed segments, which then can lead to the accumulation of drusen and macular degeneration. So that's sort of the big picture of all of the bad stuff that 7-keto cholesterol can do. I can just keep going, or if people want me to pause for questions along the way, I can, but some of the stuff that I just gave as that overview will get delved into in more detail in the next 3 figures.
Just a quick question there. Okay, maybe the terminology was throwing me off a little bit. So where's most of the negative 7-KC derived from? Because I know it was mentioned that it's endogenously formed through reactive oxygen species within the cells, and then it was mentioned that it bioaccumulates, and I was under the impression that that meant that it was, at least definition-wise, coming from the environment, that you were ingesting it and it was accumulating. So where is most of it coming from that is actually accumulating in your cells? Is it coming from environmental sources, or is it mostly being produced through just your normal everyday metabolic functions within the cells?
That's a great question, Oliver, and it's not a trivial question or a perfectly answered question, but I think I can give a pretty good answer to it. And the short answer is that it's being produced in your body, in your cells, and that's where it causes most of the problems. The somewhat longer and nuanced answer to that is that you can eat 7-keto-cholesterol, and for the most part, that's not dangerous. 7-KC is known to the FDA, to the food industry, such like that. 7-KC is not, it's not a new molecule. I didn't discover it. It's been studied since the 1940s, and it is not seen as a toxic food product. The reason is that it doesn't really get absorbed and taken up into your cells very well. What happens is that you do absorb it, but it immediately makes its way to your liver, where it is very quickly processed. The liver is the one place in your body where 7-KC can be mostly efficiently metabolized, and so from there it gets recognized as useless garbage very quickly, and will get either tagged for excretion or broken down in a couple of steps and then excreted. I do speculate in the paper that there are circumstances in which food sources of 7-KC could be dangerous, and that is not well studied enough yet. And there are examples in the literature in animal models of animals being fed large amounts of 7-KC when they have compromised ability to metabolize cholesterol in ways that are supposed to be mimicking what happens in humans when they have atherosclerosis or a genetic disease that affects their ability to metabolize cholesterol. And in those cases, you do actually see some problems from eating 7-keto-cholesterol. And another area of speculation is that, of course, it has to pass through your digestive tract before it gets taken up, and that it is speculated that it may be dangerous for people with intestinal problems, say people with IBS, and such like that. But this is an area that needs a lot more research to be done. But the main point that I want to come back to is in diseases like atherosclerosis and Alzheimer's disease and macular degeneration and other aspects of aging that are influenced by 7-KC that we haven't yet discovered, that it's probably mostly the 7-KC that is being produced inside of cells. Now, there's yet another source of 7-KC that I didn't mention, which is from oxidized cholesterol. So if you have oxidized LDL that's floating around in the bloodstream, that can also be a source of 7-KC that can be taken up by cells like macrophages and in the liver. And in that case, it tentatively seems like that 7-KC is less dangerous than the 7-KC that is produced inside of the cells, in situ, if you will. And that when I say bioaccumulates, that's what I'm talking about, is that it's being produced, and it's not that it's being produced and it's running down the food chain, but that it's being produced and it's not being cleared out, and that it does move around inside of the cell. So if you can think of, say, a 7-KC molecule being produced in the mitochondria and then ending up in the lysosome, and that way it is sort of transitioning through the cellular food chain. But perhaps the terminology of bioaccumulation isn't a perfect analogy here.
And perhaps you're going to get into this later, but it does seem to be once it accumulates, it seems to be a recalcitrant molecule, and it's are there any known enzymes that are endogenous to mammalian cells that can turn over or convert 7-KC into less toxic byproducts, or is that, or that's just really inefficient and we're looking on it? You're still looking for enzymes that can do that more efficiently?
Yeah, so it's actually something that Sense Foundation has worked on in the past, mostly looking for non-human enzymes. But it turns out there are human enzymes that can do it, and for whatever reason, they are only essentially, to our knowledge, and it has been somewhat well studied, are mostly only expressed in the liver. If you take in the lab one of those liver enzymes and artificially express it, say, in an eye cell, in one of those retinal pigment epithelial cells, and you artificially express one of those liver enzymes, you can get export of a 7-KC molecule out of the cell. But that doesn't seem to be happening naturally pretty much at all. So there are potential transgenic approaches, either using non-human or actually human enzymes and trying to force them to work in other cell types. But then you're not really plugged into the same detoxifying and excretion system that the liver already has in place. So that whole idea of engineering a resistance or a remediation of 7-KC enzymatically is still pretty theoretical in that regard. So let me talk, just show briefly the chemistry of how 7-KC occurs. You have one of a few different, or any of a few different oxygen radicals, pretty much most of them that can directly or indirectly, well, all of them through multi-steps, result in the creation of 7-ketocholesterol. So you can have a direct reaction of a hydroxyl molecule. I don't know if you can see my pointer, that if you have an OH group and it reacts with that 7 position on cholesterol that we've highlighted there, that you can create a 7-hydroxycholesterol, either 7-alpha or 7-beta, and that those can then turn into, they can take another oxidation step and go to 7-ketocholesterol. You can have peroxides that can form something similar, a 7 peroxide, which then can, in a couple of step process, turn into 7-alpha or 7-beta hydroxycholesterol. Or you could just have oxygen in the presence of a suitable catalyst or a superoxide that can react with the 7 position in a couple of different steps, end up with the same kind of peroxide, 7-ketoperoxide molecules, which can then rapidly turn into 7-ketocholesterol. Now, the main point I want to make here is that molecules like 7-alpha hydroxycholesterol, while reactive and somewhat dangerous, are not stable molecules. So if you have a 7-alpha hydroxycholesterol, it's a pretty transient molecule. It's not going to hang around. It's going to either react with another molecule and do some kind of direct free radical kind of damage, or it's going to get further oxidized into the more stable 7-ketocholesterol. So 7-KC is not a free radical. It is the product of a free radical. It's not a very reactive molecule, but it is a biologically active molecule. So it's pretty stable once you have it, and the steps that lead up to it are not stable. And that's why you tend to end up with 7-KC once you start the oxidation process. I hope that makes some amount of sense. And if anybody has any questions about that, grab me and we can go back to it. And I'll do my best to answer them.
I did have one question, just a quickie. One thing that wasn't necessarily clear from reading the various literature on the topic was the lysosome. Can it at all break down 7-KC, or is it a case of it does a little bit? What's the situation there? Because I'm getting conflicting information. Some people say it can completely, it can't do it at all. Some people suggest that it might be able to. So is the lysosome any good at breaking it down at all?
So My understanding and position is that generally it's not, with the exception of in the liver, in the presence of the liver enzymes. So there are papers that we can find where you can sort of trick the lysosome to process 7-KC, but that it seems that it's not done endogenously normally by, say, a macrophage or an eye cell, or possibly other brain cells or muscle cells, but those haven't been well studied.
So it's a definitely maybe then.
It's being very inefficiently processed in 99% of your cells, and at worst it's effectively not being processed at all.
And then the other obvious question for me anyway is, has anybody actually looked at the rate of processing this waste in old versus young cells? So in other words, can young cells metabolize 7-keto more readily than age cells?
Yeah, it's the short answer is it's not known and it's not well studied. It's a very difficult question to answer because, as I think many of you probably know, if you take cells out of an old person and start growing them in the lab, they often start acting in a lot of ways like young cells. So in that sense, like doing the experiment in the lab in just isolated cells often ends up being less meaningful. You also want to focus more on cells that aren't replicating frequently and thus diluting the accumulation of any kind of damage, whether you're talking about 7-ketocholesteryl or mitochondrial DNA damage. When you have highly replicating cells, you'll tend to dilute out that damage and have a hard time seeing the effects of it in highly replicating cells in the lab. Now, the problem with studying and answering that question in vivo is that 7-KC is fairly difficult to detect and measure. It's not impossible. We do it. Other labs do it. It's been done for decades. It's difficult, it's inefficient, and the accuracy and the sensitivity of your results is a little bit rough. So if you want to, try to quantify the amount of 7-ketocholesteryl in a particular, cell in vivo, it's difficult to do that by dissecting a mouse and staining its tissues, or just taking a small amount of tissue and measuring it. If you can get a good amount of tissue, say if you could hand me an entire plaque from a patient, which we're working on getting access to that material, I can measure the amount of 7-KC inside of it, tell you how much is there, and then in theory you can treat it with different things and try to remove the 7-ketocholesteryol from it. But it's a little bit difficult to answer your question. It is something that we're developing technologies that we hope will be able to help us answer more of in the not too distant future, including potentially diagnostic materials. But that's just between you and me and everyone else on the call.
So half the internet then, Oki. Because we're in Facebook land at the moment. So yeah, so probably half the internet before the end of the week. But yeah, oh, great answer anyway. But you can see where I'm going, because that's what proponents of programmed aging might say. They might say, well, how do we know that younger cells can't process it, which is often a popular refrain of people who subscribe to the idea of programmed aging versus damage accumulation. So it was worth asking.
Let me say something about that in the context of this next figure, which is the formation of a atherosclerotic plaque. So we chose to focus to a large degree in this paper and in our own research on atherosclerosis because we think that there's the closest to a one-to-one relationship between the accumulation of this particular damaged molecule in this particular disease, as opposed to, say, Alzheimer's disease, where I think most people would agree that there's the strongest correlations between things like tau and A beta. And if I try to come in and say, well, no, actually 7-KC is the real cause of Alzheimer's disease, you know, I'd probably get laughed at a bit. That's not to say that it's not associated with Alzheimer's disease and not important, but that it would be, I think I would have a difficult time convincing people, rightfully so, that that is like a root cause. But I think I can do a better job of painting a picture for atherosclerosis like that. The idea here is that here is an artery going across the screen from left to right, looking at what do you call that, a lateral section of the artery, and so say the blood would be flowing, say, from left to right. You have a lesion that starts in the artery, a lipid lesion, and then monocytes will be recruited to the site of that as damage, and that's what monocytes do. They get recruited to, whether it's a bacterial infection or a plaque lesion or other kinds of damage, even wounds, they'll get recruited to the site of the insult, and then invade the site of the insult. So go into the tissue, in this case, into the intermedial space of the artery, and differentiate into a macrophage to start trying to address the problem. And our model here is that the monocyte will the macrophage will then just start eating everything in sight. So they're going to try to eat up all the lipids. They're going to try to eat up all the dead cells, all the necrotic tissue there. Then, as they eat up a bunch of that, some percentage of the stuff that they're eating is 7-ketocholesterol. As the 7-ketocholesterol makes its way to the lysosome of the macrophage, the lysosome will start to shut down, and they'll lose their ability to effectively metabolize the stuff that they're eating. But they won't stop eating. They'll just keep eating themselves to death, essentially, and ballooning up into what's been known for many decades as a foam cell. And the foam, styrofoam on high resolution microscopy in the middle of the plaque. So that's our model for how 7-KC contributes to the formation of the plaque, that it forces the macrophages to go from part of the problem to being part of the solution. Any questions on that before I go on to another aspect of plaque biology and the role that 7-KC plays in that?
I don't know. You've been pretty thorough so far, Oki. Anybody else got any questions? It's fascinating. Anyone else? Oh, I did have one question about macrophages. You mentioned microglia earlier. So it's a case that 7-KC gets sucked up by not only mobile macrophages, but also the tissue resident ones, microglia. How many other cell, like tissue resident macrophage types do we know? You mentioned the eye. Are there any others that do it, or would you think personally, would you think that most macrophages would suck it up?
Yeah, I would think that macrophages could suck it up. Now, the issue though is that macrophages, once they're done doing their job, say at the site of infection or injury, they will tend to then die and get excreted from the body. So if they don't have, the plaque is in one way unique in that there's not a good method for them to get cleared out after they've, because they don't end up succeeding at their job, they fail at their job in that case. In the case of an infection, they may, eat up a certain amount of 7-KC, but then they're going to tend to get turned over and excreted pretty readily. So I can't think of other specific examples in which macrophages per se would be accumulating in other tissues, but I'd love to have examples pointed out to me in which that could happen.
But yeah,
but there's certain other cell types that can accumulate 7-ketocholesterol.
But obviously, macrophages are very keen on gobbling up what they see as junk. So I'm going to go out on a limb and say that probably most forms of macrophage, tissue resident or otherwise, will probably do that, which is it's a problem, isn't it? It's a big problem. So let's get rid of it, but more on that later.
Okay, so in the next figure, and it looks like the figure that's currently available from the journal is really crappy resolution. So if I could actually switch to a different screen, I can show you a nicer version of that. Can everybody see this version of the figure?
Wow. Yeah, that's way sharper.
Yeah, so I apologize. You go through these different steps in the publication process, and you have different versions of your manuscript that go through many different layers of editing, and it seems bureaucratic, and it is. And so unfortunately, the current version that's online, despite the fact that they had high resolution versions submitted to them for purposes of making things smaller and easier to move around, they stick a low resolution. They make my nice pictures worse before they make them better again. So here's the nice version of this figure, which is a bit more theoretical than the last figure. So the last figure basically told the story, the 7-KC aspect of it being our story, but the idea of how a plaque forms being, fairly canonically and well accepted. So now we're talking about the same kind of artery here at the bottom. So then we zoom in on inside of the wall of the artery where the calcification of the plaque happens. And this is something that's a bit more theoretical, but it's a story that I think is coming together, that we pieced together in this review article from some articles that have been published in the last, just in the last couple of years, that I think are starting to tell this story. So people used to ask me, well, if you get rid of 7-KC, is that going to do anything about vascular calcification? And I would say no. Really, we're just looking at the core bulk of the plaque, and the stiffening of the arteries caused by vascular calcification or other aspects of stiffening are unrelated. But now I'm starting to change my mind on that and think that there is a mechanism for which this occurs. And that seems to be due to the stress response that's caused by 7-ketocholesterol inside of the cells. And the part that we're piecing together here is that it's been shown that 7-ketocholesterol can induce a stress response. And it's been shown that through certain signaling pathways like PARP signaling, that can lead to the downstream calcification through inorganic phosphate and through the excretion of calcium-loaded vesicles. And it's been shown that 7-KC can induce the formation of these stress response pathways like the PARP pathway, which we didn't explicitly point out here, but we point out that it's part of the PI3 kinase pathway and ALP. What hasn't been explicitly put together is that it is in fact 7-KC that leads to the particular pro-inflammatory signaling that leads to the calcium-loaded vesicles and the apoptotic bodies that are causing the calcium calcification. So what I've put together here is a theoretical model, sort of connecting all those dots ourselves from different things that have been published in the literature from those different pathways. So we're theorizing a method by which 7-KC could be causing calcium calcification of the arteries. That may have been a bit disjointed and clear as mud, but I hope that kind of makes sense to some of our viewers.
I'm sure they'll soon pipe up if it didn't. But fascinating stuff. They are usually a lively bunch. So if anybody does have any questions, who are watching, anybody watching has questions, do feel free to join in and ask them, and we will get you the answers, or at least try to anyway. Sometimes we don't know, but we'll give it a shot.
And so there's a lot of sections. If people have read the paper, there's a lot of sections to the paper that weren't covered in the figures. So that's what makes a review article different from a primary literature article, is that you don't like have a figure for every point that you're trying to make. Sometimes we just have sections about the liver, about neurodegenerative disease, that aren't explicitly covered in a figure. And I'm happy to field questions, either general questions about broad organs or diseases and the role that 7-KC may or may not play in them, and anything else that we cover in the paper as well.
Well, I know Sven's got a very specific question, which he told me, so spoiler alert there. But, I've got a pretty obvious question, and I'm going to ask it because it's probably what everybody at home is thinking. We see what the problem is. How are we going to fix it?
Yeah, so about 8 or 9 years ago, I went to a conference on free radicals in biology and medicine, and somebody gave a presentation on 7-ketocholesterol in which they talked about all kinds of bad things that 7-KC did. And I can't remember who the speaker was, but I probably referenced one or more of their papers here in the 220 or so references that we have in this manuscript. And I went up to her afterwards and I said, wow, sounds like this 7-KC stuff is really bad. And in fact, Sen's Foundation, I was already a part of it, and I knew that we had a longstanding interest in 7-ketocholesterol and why it was bad and how it was bad. So I went up to this particular researcher on 7-KC and asked her, so what can we do about this problem? And she said, oh, I don't know. I think maybe eating a Mediterranean diet would be a good thing. So that's true to an extent, that if you have a healthy diet, you will probably accumulate, you'll have less cholesterol and therefore less of one of the steps of this reaction, right? So if you have just a simple two-step reaction, A plus B leads to C, classical chemistry, if you have less cholesterol, you're going to tend to drive that reaction less. And if you have a less oxidative environment, you're going to tend to drive that reaction less. And so eating a bad diet high in unhealthy fats and cholesterols and other bad things that produce an oxidative environment, then you may produce more 7-ketocholesterol. That's the sort of non-Sen's answer, sort of classical answer to the question. Or you can take the sort of current state of medicine is that if you have too much cholesterol, you can take cholesterol lowering drugs and affect the balance the same way. Now in the future, we want to actually get rid of the 7-KC after it accumulates, and you're not going to be able to stop the reaction. You may be able to slow it down with a good diet or cholesterol lowering drugs, but you're not going to be able to stop it. So how are we going to reverse that damage? And I think we've touched on one of those ways, which would be enzyme therapies that can either directly or indirectly or more generically hit 7-ketocholesterol. The way that I'm trying to do it in my new company is by developing a class of drugs that can grab 7-KC and drag it out of cells and out of circulation and get it excreted. And as we pointed out, if we can get it either excreted directly from the body because it's been encapsulated in something soluble, then you can urinate it out. Or if you can get it to the liver, at least a reasonably functioning liver, can handle 7-ketocholesterol pretty effectively.
It sounds like a plan, but yeah, well, obviously
I'd be happy to digress into the work that my company is doing, but I didn't really want to make this about a sort of self-promotional, self-interested pitch on my company, and sort of wanted to focus on the the biology of this nasty molecule and why it's so important.
Well, I mean, I don't think there's any issue with exploring, possible solutions, including what you're doing as a company. I'm sure the viewers at home would be keen to find out what the answers might be. But Sven, didn't you have a very specific question?
Yes. So first of all, I want to point out I'm working myself on C. elegans, and that's where my question comes from. So I'm particularly wanting to know if there is any research you have ever heard of that suggests that when you give 7-ketocholesterol to C. elegans, that it might be toxic for them.
Yes. I'm trying to remember, and I can't remember if I mentioned this if we mentioned this in the article here as it stands. Okay. So I referenced a review article here towards the end in section 5, where there's another review article, coincidentally, at the same time that we were writing this article, somebody else was writing their own review article on the related topic of not exactly on the same topic of us, but they were writing about 7-ketocholesterol and other 7-hydroxycholesterols. And they talked a lot more about model systems. And they basically theorized that they thought that C. elegans would be a good model for studying 7-KC in aging, but they didn't really provide a lot of evidence or show data that it either accumulated in the short life of a C. elegans, but I think they suggested that dosing C. elegans in some way, either in their food or I don't know, maybe injecting it into them or something, could be a good way to study the biology of 7-ketocholesterol in what is obviously a good aging model, in a short-lived aging model.
The reason why I ask is that so when you grow nematodes, you always add cholesterol to the medium because they need a source of sterols. C. elegans doesn't need cholesterol itself, it needs some other sterols, but it can synthesize them from cholesterol, and cholesterol is an easily obtained sterol. So you add cholesterol to the medium. But one thing I was thinking is that maybe some of the variation you see between experiments, between different labs, might be because some lab uses cholesterol that is more oxidized than another lab, and this then causes toxicity effects in the worm, leading to experimental problems. So that was my thinking. And in that regard, I also have a very short second question, and that is, when you have cholesterol in a solution, we always use an ethanol solution. When you store that at room temperature, how quickly does it oxidize and become oxidized cholesterol?
Yeah, so in the first question, I don't know, and I'm guessing it's not known, how C. elegans might metabolize 7-keto-cholesterol. So if they have great methods of metabolizing sterols sort of independent of their size and shape, and you just use cholesterol as a sterol, as sort of a generic sterol source for them, then it may be that 7-keto-cholesterol would just get used the same way that cholesterol does, and they wouldn't have to worry about it. So I don't think it's known well enough yet. If it did get intercalated into their membranes, I think they might have a problem, but you said that they don't actually, they must use cholesterol in their cell membranes. They're animals, right? So I would think that they would suffer the same kinds of problems that any other cell type, and many cell types have been studied, and 7-KC is pretty much universally toxic. Once you get over a certain amount of 7-KC in any cell, it is very toxic. So with the exception of the fact that maybe C. elegans have tricks for metabolizing 7-KC that most of our cells don't, it might be a good method. With the second question being that does cholesterol sort of self-oxidize in normal storage conditions into 7-keto-cholesterol? I don't have a quantitative answer for you, but qualitatively, I think know that under normal sort of atmospheric conditions or in ethanol or something like that, you're not going to have enough oxidative pressure to get a lot of oxidation of cholesterol happening, and so you're not going to have a lot of 7-KC happening there. So, for example, in food cholesterol, you don't get a ton of 7-KC unless you subject it to, say, like in spray drying of dairy products. So if you're making powdered milk and then you use an open flame at extremely high heat to spray dry your powdered milk or something, that you can then create huge amounts of 7-KC that way, where you have a heat source that it can work together with the atmospheric oxygen to react and produce 7-keto-cholesterol there.
Mm-hmm.
So unfortunately, I'm out of time, and so I don't think I can field any more questions from the audience. I'd be very happy if people wanted to follow up with me. They can email me at my SENS email, which I will always have, or my new Underdog Pharma email, both my first name, Matthew, my last name, O'Connor, at sens.org or underdogpharma.com. You can look me up on the websites for SENS or for Underdog Pharma, and I think I'm not that hard to find. And of course, everybody at Leaf knows how to get in touch with me very well. So I'm sorry to have to cut things a little bit short before we can go through absolutely everything, but I really appreciated the chance to be here and talk with you all today.
Yeah, thanks for taking
All right.
the time.
Awesome.
Thank you. Have a nice.
Thanks, everybody.
Thanks, Oki. And hopefully next time we could look at other small compounds that accumulate in aging, because I think this is an interesting avenue to explore. Not only the mechanisms that maintain longevity, but also the detrimental things that that accumulate and cause aging to happen. So with that, thanks everybody, and I hope everybody has wonderful Christmas, New Year's, Kwanzaa, Hanukkah, happy holidays.
Happy holidays, everybody.
Happy holidays. Bye-bye.
Bye.
Yeah, thanks for everybody joining us, and we look forward to well, it'll be a new Journal Club, new year, so we'll be back probably towards the end of January, I would imagine. Yes, it does seem a while away now, so everybody stay safe.
And use fresh eggs in that eggnog to avoid the 7 keto cholesterol.
Yeah, we don't really have that over here, Oliver. Although I think sometimes you can get eggnog now, but it's quite. I've never actually had it, so
I've had it a few times. It's actually very good. It's like a creamy, smooth, alcoholic drink.
I think the closest analog I could probably think it might be like is Advocar, which is basically alcoholic custard. I think it's Dutch.
My recollection is good. It's sort of a warm, alcoholic-y milkshake, basically.
Pretty much, yeah. Advocar, it is good. And if you stick it with lemonade and put a cherry in it, you get a snowball. So there you go.
All right.
Fun Christmas fact for you.
All right.
So, do try them. Do try them if you've got access to that, but obviously in moderation. As recent studies have shown that moderate, and that's the key word, moderate alcohol consumption appears to be associated with longevity. That doesn't mean you can drink 15 pints a night though, guys. So just take it easy over Christmas. And doubly so for Sven there, because I know he likes to drink, don't you, Sven?
So keyword moderation, not just for drinking, but for everything, religion and politics included.
Oh, politics and is bad for your health, so I wouldn't get involved with that. So anyway, stay safe, and thanks to everybody who joined us this evening. And it obviously goes without saying thank you to our Lifespan heroes, our monthly patrons, who make shows like this and all the other wonderful things that we do possible. If you're interested in supporting us, we would like that very much. So an ideal Christmas gift for us. So if you want to find out how to become a Lifespan hero, check out lifespan.io/heroes. So there we go. And with that said, I wish you all a very Merry Christmas and a Happy New Year. And I'm sure the rest of the Leaf team are not here do as well. So take care, all the best, and we'll see you next year.
See you, everybody. Take care.
See you next year. Bye-bye.